Okay, so we proceed as before. Should we or wait for people to join slowly? But I think due to the interest of time, allow me to proceed. Okay. So just a second. Okay, so this is what we are talking about. We said apples are tasty and cookies are good. So I was asking at this point, do we have any questions? Any questions on what we've done so far? Any questions? Or we are waiting for me to ask the question? Please go ahead and ask. Feel free to ask. Okay, so we proceed. So in DNA, there are two strands, okay, which form a double helix. That's why it is called double helix. They run in opposite directions. Okay, the base pairs are arranged they are held together by hydrogen bonding. So there is hydrogen bonding that holds these base pairs together. Okay, so in the bonding between C and G, okay, the base pairing is very specific because of the hydrogen bonds. The number of hydrogen bonds between A and T is different from that between C and G. Okay. Two hydrogen bonds are between A and T. C and G has 3 hydrogen bonds. Take note, there are 2 hydrogen bonds between A and T, and there are 3 hydrogen bonds between C and G. So these are small things which you need to not forget. Okay. So everything else is the same, apart from the number of hydrogen bonds and which pairs are together. So because of that, our DNA, each pair consists of a purine and a pyrimidine. That is why they have the same width. Otherwise, if it was purine and purine, it would be larger at one point and smaller at another point. Okay. So that's DNA. Okay. So Watson and Crick are the 2 guys who came up with this theory of it being a double helix. All right. So we can't go past through biology without acknowledging them. Okay. So DNA base pairing is antiparallel. So we are seeing the first one, which is the leading strand. We have 5' to 3'. So meaning they are joining by the 3'. Okay. The next pair is joining by the 3'. So when you look at transcription and translation, we'll understand this further. Then the other one is in the opposite direction, 3' to 5'. Okay. So they are anti-parallel because the other one is in this direction and the other one is in the opposite direction. They are parallel, but in different directions. That's why they are anti-parallel. parallel. So when you separate them, this is how they would look. So they pair or they attach to each other because of the hydrogen bonds. The phosphates at the end, nitrogenous base, the sugar, there's nitrogenous bases in the middle, the sugars in between the phosphate and the nitrogenous bases. Okay. So RNA is usually singular, a single strand. DNA is a double strand. Okay. Double strand is created by the hydrogen bonds between the nucleotides. Okay, this is just an emphasis on what we've already talked about. Okay, now we look at RNA, the structure of RNA. So it is a single polynucleotide. That's the first key. It is a single polynucleotide. Then the next is that at position 2, RNA has an OH, and at position 3, it has a hydroxyl group. So at both position 2 and 3, there is a hydroxyl group in RNA. In DNA, at position 2, there's only hydrogen. So RNA has a hydroxyl group. It has more oxygen atoms, or there's oxygen. There's more oxygen in RNA than in DNA. Okay. So there are 3 types of RNA. We have messenger, transfer, and ribosomal. The next difference we have is that RNA has uracil and not thymine. Okay. All right. So the rest. We look at messenger RNA. Messenger RNA is a single strand formed on a single strand of DNA. Okay, when we look at transcription, we understand this further. But it's just an example of the RNA. So we have messenger RNA. Okay. Then we also have ribosomal RNA, which is rRNA. So this one, it comes in different sizes. We have the 70S, 30S, 80S. Okay, 70, 80, and so on. When you look at this further, we also understand it nicely. So each ribosomal is made up of 2 subunits. So 70S has 50 and 30, which is found in prokaryotes. Then eukaryotes have 80S, which is 60 and 40. So other authors who have different variations in terms of the numbers, but remember that prokaryotes is 70S, eukaryotes have 80S. What is in the brackets may differ. Okay. Transfer RNA, which is the 3rd type of RNA. Okay. So this one is special because it has arms. It has arms. Okay. It has 4 arms. The first one is the receptor arm. Then you have the variable arm, the D arm, and the anticodon arm. So when you look at the structure here, the 4 arms, we have the receptor arm here. The receptor arm, where we have the hydroxyl group, the T arm, okay, the variable arm, anticodon arm, and the D arm. So the difference is that right after the receptor, we have the T arm. The D arm is right before the receptor arm. Okay. So that's about transfer RNA. This is how it looks. So even in your questions or in your MCQs, wherever you see something with 4 arms, it's transfer RNA. You don't even need to waste time. You just need to remember the arms. That the transfer RNA. Sorry?
I've got a question.
Yes, please go ahead.
What's the function of what's the function of D arm and D?
Okay. So we look here. So each of these, we'll discuss them further in detail when we look at transcription and translation. For now, we're looking at the structure. Okay. I hope you can keep your questions for then so that we have room for other questions. Is that okay? We can keep it when we do translation and transcription, then you see how they function properly. Otherwise, for now, we can just keep it at the arms. We look at the structure, then we look at the function later. Okay.
Okay.
All right.
All right.
Okay. So
Okay. So
we have this rRNA, which is starting from the 3' and ending in the 5'. That is another key thing to note about this arm. It is starting from the 3' and ending in the 5'. Okay. Any questions?
I have a question.
Yes, concerning this lecture, isn't it?
Yes.
Okay.
On the part for arms, I'm a bit confused because you are saying it has 4 arms, right?
Yes.
But again, when we count there is acceptor arm, T arm, D arm, anticodon arm, and the variable arm, and they're 5.
Okay. So if you look at our previous slide, okay, so there's the acceptor arm, the variable arm, the D arm, and the anticodon arm. These are the 4 arms that we are considering. So we have the D arm, the anticodon arm, the T arm, okay. So you have the D, anticodon, variable, okay, and the acceptor. So the 5th one that we are seeing, we are looking at it in terms of function. We'll consider the 4 arms instead of the 5 arms, in terms of function. That is why, like I said earlier, when it comes to transcription and translation, we'll get to explore this further. Okay. So we'll have a lot of questions then to ask, and to understand why we are saying 4 arms, despite the 5 that we are seeing.
All right.
Okay. Any other question? Okay, so we can look at the last slide, which is comparing our DNA and RNA. So the similarities is that DNA and RNA are present in all prokaryotes and eukaryotes. Okay. Then they carry genetic information. They are made of nucleotides. There is polymerization, okay, which results in formation of that strand, and they are cleaved by nucleases. Okay. Then when we look at the differences, DNA stores and transmits genetic information, while RNA just transmits genetic information. Okay, then DNA is double-stranded, RNA is single-stranded. Okay, DNA is chemically stable, meaning it can be stored as DNA. RNA is unstable, so it is just a transition state. Okay, then DNA has thymine in the place of uracil. Then RNA has uracil in the flexosymide. Then DNA is a huge molecule, RNA is a smaller molecule. There's usually only one type of DNA, but RNA has 3 types. Okay. Any questions, any additions before I ask a few questions? Any questions or additions?
So I have a question.
Yes, madam.
On the fifth similarity, can you explain more, sir?
Okay. So the fifth similarity, which is cleaved by nucleases. So that is the cutting. Okay. Whenever we have a complete RNA, there is cutting of that RNA strand. Again, this topic is only picking a very small component of the whole larger or the whole wide topic of nucleotides or nucleic acids. So when you look at transcription and translation, we're going to see nucleases and other enzymes which help in the formation of DNA and RNA. But something that is to note is that they are cleaved by nucleases. This will be clearer when we see the enzyme nucleus coming in. Cleaved means cutting. Have I answered the question?
Yes, sir.
Okay. Any other? Okay. So if there are no questions, then I think today I have no questions for us to discuss. We delayed a little, but I think in the next session, when we look at next topic, we'll discuss the questions. We'll combine them. When we look at the transcription and translation, we'll have all these questions coming in. Any additions from anyone?
Sir,
Yes, sir.
sir, I was thinking maybe we can have a maker for this one because we are too fast. I was missing some points.
Okay, I was asking if
Yes,
I was
I am
too
moving
fast.
too fast.
I was asking if I was too fast and you are quiet. Okay. So
We can interrupt you when you started already.
okay.
Yes,
So
sir.
what we'll do is we'll organize for that makeup. Is that okay?
All right, sir.
Yes, we'll combine it with the translation and
Transcription.
yes, and transcription. We'll still revisit this. I think then it will make more sense.
No,
that
all
okay?
right, sir.
Is that okay?
Yes, sir.
Yes.
Okay. So we'll meet in 8 minutes for those who are coming for the chemistry tutorial. We have another makeup on atomic structure. So we'll meet in 8 minutes. Use the same link to join. Okay. Thank you and enjoy the rest of the evening.